My name is Ross Kagan. I am the Regis Chair of Precision Medicine at University of Glasgow, which I recently moved to along with my wife, who runs COVID testing here in Scotland. I am a cancer biologist who uses Drosophila as the jumping off point for pretty much everything we do. And then we sort of move on through. I'm a laboratory that runs clinical trials. We develop drugs. One drug's gone through the clinics. We have more going through and so on. So that's my expertise. And I've had the privilege to be connected to DrosAfrica. And it really has been a privilege to meet. It's kind of cool to me that I actually recognize some of you here, which is nice. Hopefully I'll recognize more after this meeting, so please feel free to say hi. So I just want to say a few things about DrosAfrica, and I'm really the wrong person to say this because I am not the founder at all. I'm a newcomer to this. The founders, Lola Martin Bermudo, Isa Palacios, and Marta Vicente Crespo, founded this actually several years ago now. And the idea was, and I think that idea was a brilliant one, is that Drosophila is a very powerful model system. I think everybody here would agree. But it's powerful because both you can do a lot of things with it, and also because it is quick, it's inexpensive, you don't need a lot of equipment behind it and so on, which is one of the reasons I do like it as a model system. And it seemed to be a good match for a sort of young, burgeoning African science community. And so they, and I'm also going to do a shout out now for other people who have helped put together this workshop. In addition to Isa, Lola, and Marta, Hyatt, Salome, Jose Casal, Wissam Meshrif, and Luca Gebel have all made important contributions to help make this happen. And I think we're all pretty excited to have an excuse to bring in some really elite speakers. So these are some of the top Drosophila people on the planet. We also vetted them for the ability to give a good talk. So no pressure, but we expect an excellent talk. And most importantly, we are looking forward to hearing good questions from you in the audience. So fortunately, except for one exception, people who are tuning in today are not in my lab, because if you were, I would really push you to be asking questions. This is an opportunity to introduce yourself to the international fly community. For those of you for which that's important. And this is an opportunity for us to continue and build on this discussion. You know, Africa is really sort of a rising scientific group. And as somebody who believes deeply in the power of Drosophila, I'm excited to do a small part to help bring the African community more solidly into the Drosophila field, which can always use new and young minds to help. So ask questions. The way to ask questions is to get on chat here and post a question. And then what I'll do is I will ask that question to the person, or if it seems like a question that's a little bit complicated, I'll actually reach out to you and ask you to go ahead and ask that question yourself. But please don't be shy, ask questions. And also, because we are really sort of bang, bang. Let's do the mute again. There we go. Because we are having relatively short talks, they're going to be about 30, 35 minutes and then about 10 minutes for questions. Because that is really sort of bang, bang. We've opened up a Slack channel, which I think many of you have introduced yourself on. So it's actually been fun to do a little bit of chatting with you on Slack. So that's a great opportunity for the next few days and even onward to ask questions for the speakers. Whenever you have a moment, if you have time and if you so desire, check Slack and see if you have questions directed towards you. So that conversation can continue. We are recording this and we will post it as soon as I figure out how that's done. And the purpose there is that we know that, you know, with Zoom, there's going to be times where there are disruptions to Wi-Fi and so on and so forth. So we're going to make the recordings of these available to everybody. And also sometimes it helps to just hear something a second time to really stop and start and really dig into it. Now, I have created a poll just to kind of get everybody started and going. So I'm gonna have a poll each day. And today's poll, which I'm just gonna have a quick snap question, please answer. I'll probably wait about a minute or so for answers. And our first poll, which is a yes or no, is whether you've ever worked with Drosophila. So please click and let me know. The responses are coming in. This also helps us and me understand the community that we're talking to. Is it people that have worked with flies or not so much? So far it's pretty, well, now it's starting to skew. Apparently the people who work in Drosophila are faster responders. People who don't are slower but more numerous. So I'll give it a few more seconds. Okay, groovy. And so it looks like about 55% of you have not yet worked. Here, I'll share the results. 55% have not worked with Drosophila and 45% have. So it's about 50-50. So for speakers, again, understand that there's a mixed level of expertise. Cool, and thanks for participating in that in the poll. So, a couple more points. First of all, and I have been working on this a fair bit, so I expect at least a few applause lines for this. I do want to say, as I mentioned, asking questions is important because we're hoping that the next Drosophila Nobel Prize will come out of the continent. So let me say a few words about these guys here. So there have been 6 Nobel Prizes that have been won or shared by Drosophila people. That's a lot, especially for a little insect. So why have flies been so successful? Well, we're about to hear over the next 3 days. So if you're ambitious and you're expecting to Nobel and look like these guys somehow, then Drosophila is a good start. So this was a pair of Nobels, one in 1933 and in 1945. I can barely see my own notes, by T.H. Morgan and Hermann Muller. And basically what they did was to introduce Drosophila, in the case of T.H. Morgan, really introduce genetics to the biology community and showed how they can be very powerful for exploring fundamental questions of how genes drive development, now how they drive behavior, and so on. So T.H. Morgan, back really at the turn of the last century, he tried over 30 different types of animals to figure out basically what would breed in a lab. And pretty much the only thing you get to breed consistently in a lab was fruit flies. And that's actually the reason that he picked them. But there we go. So that's why he originally picked it. But in fact, it turned out to be a fortuitous move. Fruit flies have a lot of colorations and bristles and so on that can be used as markers to follow genes. And at this point, they're really set in as one of the 4 or 5 major model systems that the majority of biology labs use. So T.H. Morgan really founded that and really founded modern genetics. Hermann Muller did something very specific but very important, which is they used to have to push hundreds of thousands, even millions of fruit flies to find the occasional mutation, like one mutation. What Muller showed was that if you just irradiate the flies, you can damage the DNA and you get a much higher rate of mutagenesis. And that really was the tool that enabled saturation genetic screening and so on to come online. And so those two fellows basically were the ones that founded modern genetics and allowed it to happen. So that's my Nobel discussion for today to get you pumped up to win your own. I expect to see at least a couple out of this group. And then I want you to credit this workshop when you get it, when you're in Sweden. So over the next 3 days, tomorrow we're going to focus on flies, disease, and aging. This is something close to my heart because my lab focuses on disease, but also aging. You obviously don't want an animal that takes 80 years plus to get old. You want something that will move quickly. Drosophila is really nice for that. And we'll also hear about aspects of aging. On Wednesday, we'll go down the heart of what has really made Drosophila in the modern age, which is developmental biology. We'll hear quite a bit of developmental biology and cell biology from a number of really elite speakers. So I'm pretty pumped about that because I was trained as a developmental biologist. But today, we're going to talk about something actually that Hyatt was the one who suggested, and I think was a great suggestion. And that is Drosophila studies in nutrition, microbials, and also in toxicity and toxicology. So we have a great lineup today, starting out with Amos Abalaji, and then Adam Dobson, who I suspect is about 50 feet behind me at the moment, I'm going to guess, and Nicole Broderick and Carlos Ribeiro. So listen, thank you everybody for being part of this. I'm going to keep time, so I'm going to butt in at some point and get nasty, so don't make me do that. And please post your questions on the chat, and let's get this on.
So first of all, thank you, huge thank you to the organizers for organizing this fantastic initiative. I think it's really wonderful that we can speak internationally and really expand our professional networks, get to know people doing exciting research in new places, and it's a great opportunity to share my research here as well. So I'm going to be talking today about some work that has been going on for a few years, which I've been involved with in a few places before coming here to Glasgow to start my own lab just about a year ago. So I'm still pretty new to running my own group, but I'm going to show you the first little bit of data that we've got. So I'm interested in the basic biology of nutrition and how we can use flies to actually understand how nutrition works, not just for flies, but for all animals. And hopefully by doing that, we can learn a little bit about how nutrition actually affects human health as well. So I'll just put on my laser pointer here. Okay, so what I'm hoping is that by the end of the presentation, I will have convinced you a little bit that in order to really understand how nutrition works, we really need to understand the interaction of these three things. So diet is important for how healthy you are. Your grandmother can tell you that. But what we're finding is that to understand how diet affects your health, you also need to understand how genetics works, so the genes that a certain animal has and which are switched on or off, and also the bacteria which live in the gut. So one of the reasons to be interested in this stuff is the context of human health. Now, human health at the minute around the world is experiencing what you call a triple whammy, where first of all, lifespan is increasing. So we're basically getting better at keeping people alive for longer. So medicine and public health have improved really dramatically worldwide over the last 100 years, a couple of hundred years. What that means, though, is that there are more and more people reaching older ages and starting to develop what we might call diseases of aging. So older people are more prone to developing cancer, to developing dementia, and cardiac disease. And then at the same time as those two things happening, we also have an increased burden of metabolic disease. So things like diabetes and obesity are on the rise. So put all of these three things together, and what you find is that overall human population health is decreasing. So let's just look quickly at some data that define that. So this little video here shows us the rates of obesity around the world. So basically in each country, as the color goes darker, more and more people in that year were obese. So for example, here we can see that in 1988 in Canada, about 10% of the population was obese, flips in the early 90s. Now, you can see that there is substantial variability here around the world, but the important message here is that for every single country in the world, in the 40 years that this recent study looked at, every single country became more obese. So basically people worldwide are getting fatter and in states of worse health as a consequence. And what about aging? So we're also interested in the impact of nutrition on aging. So here is a projection, not from worldwide, but just from Europe, showing the proportion of the population over 60 years old from the middle of the last century and then projecting up until the middle of the next century. And what you can see is that basically we have more and more old people around, and this is not expected to decrease anytime soon. So it's good, obviously, to have more wiser, older people about, but unfortunately there is a flip side to this, which is that for both males and females, for these three different classes of disease, what we see is that as people age, there is an increased incidence of disease. So put these two things together, you have more people living to older ages, and then those people tend to develop these diseases of aging. So what can a fly tell us about these things, and why is it that a fly is a useful tool to understand diseases of metabolism and diseases of aging? Well, probably one of the most important features of Drosophila for this question is the principle of evolutionary conservation. So basically, this is because this idea outlines that most of the genes that you will find in a fly, you will also find in a person. And the majority of those genes work in the same way. So for example, if you look at diseases that cause genes in humans, 75% of those genes actually have a functional ortholog in the fly. So that basically means that 75% of genes that cause human disease are actually found in Drosophila as well. So we can study what they do in Drosophila and then work out what they might do in people. And even more compelling than that is that when we look at actually the functional domains of those proteins, so the bit that actually does the work of the protein, 85% of those sequences are actually conserved between humans and flies. So this means that we can actually study what these proteins do in Drosophila. We can also study what they do in mice, in worms, and many other organisms. And that means that we can actually get a good grasp of what those proteins do. So here's an example close to my heart of insulin signaling. Now, I'm sure that this is a topic that you'll hear very much more about in Professor Partridge's talk tomorrow. So this is how the insulin signaling pathway works in Drosophila. So we have these peptides that circulate around, they bind a receptor, and then they signal through all these proteins and then eventually contact a protein called FOXO in the nucleus. And FOXO turns other genes on and off. But the important thing to note about this graphic here is that the pathway is very much the same in the nematode C. elegans and it is in mammals. So in nematodes and mammals, they signal through very much the same proteins, and then the same process happens at the end. So what that means is that if we can understand how this thing works in our flies, then we've got a pretty good chance of understanding what it's probably doing in other animals as well. Now, insulin signaling is an important thing for aging. So this is a trio of classic studies in the aging field, which show what happens to lifespan when you mutate the insulin signaling pathway. So if we start here with our flies, this is a plot of lifespan. So we have here the proportion surviving over time. Now, what that means is that as these curves move over to the right, then those flies are living on average longer. So that suggests that something is better about the way that they are aging. So here in black, we have flies which were mutant for the insulin receptor substrate, Chico. And you can see that they're actually significantly longer lived than the wild type. Now, this wasn't just done in this study, this has been done hundreds of times since then. And it's not only been done in Drosophila, but it was previously done in 1994 by a similar mutation in the nematode C. elegans. So we see here this really incredible over doubling of lifespan in C. elegans. So basically we're getting the same phenotype in our worm and our fly by mutating pretty much the same pathway. Now that's important because these organisms last shared a common ancestor 300 million years ago. So the same protein is having the same effect after all of that time. And so it's not surprising that we then see the same thing in mice. So again, when we mutate the insulin receptor substrate, we see this nice increase in mouse lifespan, suggesting that actually the same protein is affecting aging across all of these different animals. So we can't obviously do these experiments in people, but we can look at what the insulin pathway looks like in certain people. Now, this gentleman here is from Okinawa, and the Okinawans are a group which are really famous for their long and healthy lives. And this guy here is 95 years old, but you'll notice that he's actually planking. So he's doing this pretty intensive exercise at the age of 95. And so it's interesting to look at the genetics of groups like the Okinawans and see if we can actually find variation in these pathways which corresponds to their long life in the same way as we see in these model systems. And we don't just see lifespan. So I'm not going to show you any data here, but we actually see changes in fly mobility as flies age as well. So for example, they get less good at climbing as they age. They get a bit slower and a little bit lazier. We can actually rescue these phenotypes through the same tools by mutating certain pathways. So that tells us a little bit about genetics, but obviously as I've explained, I'm interested in diet as well. So how can we manipulate fly diet? Well, flies feed very happily on diets that consist of just yeast and sugar. So this is what we feed them in our lab. So the yeast component consists of the protein and lipid and micronutrients that our flies need. And we also feed them on sugar, which obviously is sugar, but this is the major source of carbohydrate in the diet. So that's good. We have these different nutrients that we can manipulate. But the cool thing about these two nutrients, and a very lucky thing in terms of studying Drosophila, is that the calories that you get from a gram of yeast are about the same as the calories that you get from a gram of sugar. So what that means is that you can actually manipulate not just the levels of calories in the diet, but you can also manipulate the quality of the diet by changing the ratio of yeast to sugar. So some colleagues of mine a few years ago looked at what that does to lifespan. So here again, we're looking at the proportion of flies that survive over time. Now, remember that yeast and sugar have about the same caloric value. So in blue here, we have a low calorie diet, and in red, we have a slightly higher calorie diet. Now, what they did here was to keep the calories about the same, but they skewed the ratio. So basically in the dashed lines here, we're actually reducing the amount of yeast. So what you see is that as you reduce the amount of yeast and increase the amount of sugar, you see this increase in lifespan. And when you do that at a higher calorie level, you still see the same effect. So what that means is that yes, there is an effect of calories on lifespan. So calories overall are bad for your lifespan. But even when calories are the same, you can actually reduce the amount of yeast, and then you get an even further extension of lifespan. So it tells us that actually there is an effect of the quality of your diet on lifespan. So that tells us about what goes on 4 different diets, but we can actually look over an even bigger space of diets and then compare what happens between flies and other animals. So here, so it turned out that the important part of the yeast is probably protein. So here this is plotted as the amount of protein that a fly eats per day, and here the amount of carbohydrate that a fly eats per day. And by looking across many, many diets, they could actually start to understand this quite complicated picture of how lifespan responds to diet. And so all you need to know here is that the hot colors correspond to a long life and the blue colors correspond to a low life. So we see that on these high protein, low carbohydrate diets, then they have a fairly short lifespan. And then as we get up to these higher protein, lower carbohydrate diets, we have a longer lifespan. Sorry, I should have said that correctly. On these lower protein, higher carbohydrate diets, we actually have an extension of fly lifespan. So you can see it's a fairly continuous effect of the ratio of protein to carbohydrate. Now, this is a complex way of looking at it, but this is really the state of the art in understanding how diet affects various different traits, and it's been very well applied to lifespan. And it's not just been done so in flies, but also in mice as well. And in mice, the key thing is that you can see the same picture. So again, on these high protein, low carbohydrate diets, you see a relatively short lifespan. And then as we move up into these low protein, high carbohydrate diets, we see a significant extension of lifespan. And you can see basically the same picture is happening here in mice as we see in flies. But the key thing is that this study was done 6 years before the mouse work was published. So it really tells us that we can make foundational discoveries in Drosophila about the way that nutrition works, which then guide the way that we do subsequent experiments in mice and other organisms to really understand basic biology. So that tells us a little bit about diet and aging, but we're also interested in obesity and these other processes. Now, it's fairly well understood that certain diets will be better for your levels of body fat than others. So if you eat a healthy diet full of vegetables and lots of natural foods, you'll tend to do fairly well. Unfortunately, if you eat a diet with burger and cake and so on, then you may gain a little weight as a consequence. But can we actually understand this process using Drosophila? So again, we manipulate yeast and we manipulate sugar, and we can actually look at how the levels of these two, both on their own and together, affect affect obesity. Now, this is a similar plot where we're looking again at the level of sugar in the diet on the x-axis, and here on the y-axis, we have the level of yeast. And you can basically see the reverse picture of what you see with lifespan, that on high sugar, low yeast diets, then we see a very high level of body fat. So this is measured as triglyceride here. So high fat on high sugar and low yeast. And then we get onto low yeast and high sugar, then we see quite a low level of fat. So this tells us that you can actually use flies to uncover similar processes in the metabolism of fat as you see in humans. Now, the last thing that I just wanted to introduce is this idea that it's not just the food that you eat, it's also the bacteria which help you to process those nutrients. So there's been a real explosion of interest in the past 10 years or so in the microbiome. So these are the bacteria which live in guts. So here, this issue of Nature was all about the human gut bacteria, but it turns out that these are actually found throughout animals. So for example, in humans, when you look at the total number of cells in the body, then about 50% of them are actually human, but around about 50%, sometimes slightly over 50% of the number of cells that are in your body are actually bacteria, and these are predominantly bacteria that live in the gut. So these are, cells clearly actually have potentially a large role to play in your health because they're sat there in the gut, and any food that you eat will actually be potentially processed by these bacteria. Now, it's not just humans that have gut bacteria, flies have them too. So this is a nice picture from a recent paper a couple of years ago. This is freely available online. And so this is a graphic of a fly gut. So the head would be around about here, and this would be the back end of the fly. And what we see if we actually zoom in on certain areas, so here just looking at this region of the gut, is, so in blue here, we're looking at DNA. So these are largely fly nuclei. And in red, we're looking at bacteria. And you can see these tiny little red spots. If we zoom in even further, then you can actually see that there are bacteria which are associated with the inside of the fly gut. And this mirrors what we see in humans. So we're really interested in using this feature of the fly to understand the impact of bacteria on their metabolic health. And this is something that you'll hear plenty about this afternoon. So that's kind of the introductory part. So I'm just going to show you some data that we've gathered over the past few years that hopefully will convince you that if we understand how diet and genetics and microbes work together, then we can start to predict what might be a good thing to have in order to reduce obesity and to improve healthy aging. Now, we don't have all of the connections here yet, but I'll show you some of the kind of one-by-one connections that we've managed to make so far. So first of all, let's think about these gut bacteria, these microbial symbionts that live in your gut and provide nutrients. What do they do for obesity? So these are some data that we gathered a few years ago in Drosophila. So what we're looking at here is what happens when you either have microbes, here with a plus, or when you eliminate microbes, here with a negative. And so what we measured here is body weight. So this was dry mass. We measured protein, and we measured triglycerides. So this is body fat. And so what you see is that when you remove the microbiota, then our flies are underweight. They have a reduced density of protein, and they also suddenly gain this enormous increase in the levels of body fat. So what this tells us is that basically our germ-free flies, which we can also call azenic, these germ-free flies are a bit messed up. They are not in a good metabolic place, and these are actually some of the hallmarks of metabolic syndrome. This doesn't just happen in our fly, we can see very similar things in mice. So on the left here, we have a mouse which was raised with a normal, healthy microbiota. And on the left here, we've got a mouse that was raised with a perturbed microbiota. And what you can see in this kind of gruesome picture is this enormous deposit of visceral fat in the mouse with a disturbed microbiome. So it tells us that microbes aren't just important for metabolism in flies, but also these effects are conserved as well in mice. So that tells us a little bit about how we can start to fill in this triangle and the role that bacteria play on their own. But do the bacteria have an influence on obesity which interacts with this effect of diet? So to put that another way, does a change in diet depend on a change in the bacteria? So this is the first part of a more complex experiment. So let's look in at a simple level and then we'll zoom out to the big picture. So here, if we look just on a diet where our flies were fed 100 grams per litre of yeast and 25 grams per litre of sugar, what happens when we eliminate the microbiota? So we raise our germ-free flies once again, and just as we saw a minute ago, those flies have an increased level of body fat. So does that change, that impact of the microbiota, depend on the diet that the fly is being fed? So if we look now on a slightly lower level of yeast, what we see is that actually there's very little effect of that yeast on the level of fat in our conventionally reared flies. But in our azenic flies, then suddenly the effect of removing the microbiota is even worse. So this tells us that the metabolic effects of microbiota depend on yeast. So the impact of removing bacteria on fat depends on diet. But we didn't just look at 150 grams of yeast, we actually looked at 225 here as well. And you can see that actually there's a fairly complex picture that the impact of the microbiota and obesity depends on the yeast levels of the diet. So it's not just important here to think about microbes, you really need to understand what's going on the diet. Now remember that fly diet consists not just of yeast, but we can also manipulate the level of sugar in it. So what happens when we increase the amount of sugar which is in the diet? Now, if there was no effect of sugar, then we'd expect to see the same thing here. So that would tell us that there is an effect of yeast and of bacteria, but not an effect of sugar. We might alternatively see that we see the same kind of shape of these curves, but they're suddenly just boosted up, which would tell us that there's an effect of sugar, but it's independent of the yeast and independent of the bacteria. But actually, we didn't see either of those things. What we see is that not just the impact of yeast is important, but that the shape of these curves is actually different. So this tells us not just that the bacteria and the yeast are important, but the way that the bacteria and the yeast interact depends on the level of sugar that you have as well. So it's really consistent with this idea that both quantity and quality of nutrients in the diet will influence the way that microbes affect your body fat. So we didn't just do this on our 8 diets, we actually did this on a full set of 16 diets where we looked at all of these different levels of yeast, we looked at the same concentrations of sugar, and we measured body fat with and without microbes on all of those different levels. And so you can start to see this quite complex picture emerging where the actual relative amounts of dietary nutrients and the bacteria interact to determine body fat. And we didn't just do this for body fat, we also did this for protein content and for body mass. And you see the same thing. Now there's a lot to take in on this graph, but the important thing to take from this is this message here, that the metabolic effect of microbiota depends on the yeast and the sugar and the yeast to sugar ratio in the diet. So these 3 things are actually codependent, and you need to get all 3 right in order to get your metabolic phenotype as healthy as possible. So that tells us that the gut bacteria and the microbiome are interacting in flies to determine obesity. Tell us a little bit about nutrient quality, but that's not the only thing that we can vary here, right? So we've seen that there is an effect of sugar. Now, in many experiments, we will feed our Drosophila with sucrose. Now, sucrose is a molecule which consists of 1 molecule of glucose and 1 molecule of fructose. Now, these are the data which are the first data that we have from my lab here in Glasgow, and so I'm really excited to share them. This is the first time that I've actually shared these data in public. So I was interested to ask, does it matter which sugar you have? Do you have a different effect of glucose than you have of fructose? And if that was the case, it might tell us that our microbes are actually involved in processing these nutrients and turning them into something that we can use. I say we, obviously mean our flies. But that could be really useful information because fructose specifically has been very strongly implicated in these increases that we see in global human obesity. And it's especially the case because it's often used as a sweetener in Coca-Cola. So fructose tastes really sweet. So we asked, does the effect of the microbiota change if you feed flies on glucose relative to if you feed them fructose? So I don't know what's going on with this green thing here. So I'm just, there we go, that's gone. All right, thank you. All right. So this is what happens when we look at, first of all, body fat, when we feed our flies on glucose. So we see the same picture as we see before, that when we remove the microbiota, then our flies get fatter on a 5% glucose diet. And the flies also lose a little bit of weight. So this is a consistent picture. If we feed the flies on fructose rather than on glucose, what happens? Now, if fructose was just worse than glucose and the bacteria was somehow processing this, then we might expect that we just see something like this, where the overall level is increased and the body mass is decreased. But actually we see that there's quite a dramatic change. So rather than our germ-free flies suddenly being lighter in terms of their body mass, on fructose they actually suddenly get a lot heavier. And that's because it seems like when the flies are fed high fructose diet, having the microbiota is really bad for your body mass. You suddenly lose a lot of body weight. And we also see this loss of triglycerides. So they're losing even more triglycerides when they have bacteria than when they don't. So it tells us that there's this interaction, not just in terms of bacteria and amount of sugar, but also which sugar you have. And so I think that's really cool because these sugars are not, their atomic formula is the same. So you have the same number of carbons, hydrogens, and oxygens, but you have a very different effect, and that depends on which bacteria that you have around. And that even interacts with the level of the sugars that you've got. So what we're seeing here is a picture that for obesity, gut bacteria and diet are actually interacting. So our azenic flies are fat, they're underweight, and they have a low protein density. But that we really need to think about diet here as well, and that the quantities of yeast and sugar change both body fat and protein density and body mass, and that's depending on the bacteria. So there's a really strong interaction of these things that determine your overall health. So we're starting to fill in this picture now. We're starting to see that our microbes and our diet interact to regulate your obesity, the body fat. But what about bacteria and aging? Do they interact? So here are some data from a guy who's recently joined my lab, Dave Sonino. And so what we're looking at here is the lifespan of flies, so survival over time, when they're reared either with bacteria here in black or without bacteria in red. And what you can see is that germ-free bacteria are actually living longer than those that have bacteria. And this isn't just a fly phenomenon, because if you go back through the literature, you can actually find a number of studies which show the same thing in mice. So this was actually first shown in 1966. And basically here you can see that these mice that were reared without bacteria are significantly longer lived. So we're seeing the same thing in our flies as we're seeing in our mice. So that fills in another part of this picture, right? It tells us that our gut bacteria are influencing aging as well. But do diet and genetics also affect aging? So in the long term, I'm hoping to really fill in this triangle. We don't know about the role of genetics or diet in how gut bacteria affect aging, but we do know about how these two affect each other. So let's have a look at those data. So one of the best established regulators of aging is the insulin pathway. So this is a series of proteins where, as I showed you right at the top of the talk, these proteins actually affect aging, and they do so not just in flies, but also in mice and in worms. So we can use our really cool genetic tools in Drosophila to mess around with this pathway and see if that changes the way the diet affects lifespan. So a series of proteins which are now thought to be part of this pathway in the RAS pathway were things that we messed around with here to see if they change the effect of diet on health. So what we did was to overexpress this protein pointed. Now this is the ortholog of a human gene called ETS1, and not so much is known about what it does in adult animals. So we looked at what it does with respect to body fat and to aging. So when we overexpress pointed here in red, on a low sugar diet, what we see is that not really very much happens for aging here at all. Now, in some of the figures I showed you previously, you could have pulled out that sugar is bad for lifespan. And that's exactly what we see here. So when we go to these solid lines here, this represents aging and survival when flies are fed a high sugar diet. But the important thing to note here is that when we're overexpressing pointed, then suddenly that actually seems to protect our flies against this toxic effect. And we see a similar thing with body fat as well. So when we feed our flies on a high sugar diet, again, they gain weight. If you do that at the same time as overexpressing pointed, which should basically turn the level of this pathway up, then actually that is defending our flies against this obesogenic effect of sugar. So this tells us that we can actually interfere with the way that diet and signaling work in such a way that we can actually defend ourselves against some of these negative effects of a poor diet. Now that wasn't just relevant for the kind of long-term effects of sugar, but we were even able to show that these pathways and diet interact on a long-term scale as well. So what we did here was to feed our flies with a high sugar diet for just this early period in life. So we fed them a high sugar diet for 1 week or 2 weeks or 3 weeks. And then what we did was to restart our measurements of survival. So all that we're measuring here afterwards is not the effect of feeding on sugar, it's actually the effect of having previously fed on a high sugar diet. So we're just measuring the effect of dietary history. And what you can see here, for every condition where the flies were previously fed on a high sugar diet, they actually start dying earlier. So this tells us that it's not just the level of sugar that matters, as we've seen here, but actually your previous feeding on sugar can even have long-lasting effects on how well you survive into late life. So again, we asked, do your genetics matter for this? And does the signaling through the insulin pathway matter? So here we did a similar manipulation. Instead of regulating pointed here, we actually mutated this particular gene, FOXO. So here our FOXO mutants are in these dashed lines. And so we manipulated sugar here in a number of different ways, but in every case in the solid lines, what you see is that an increase in early life sugar actually accelerated mortality. So the flies represented here in red are dying earlier. But when you knock out this gene FOXO, you don't see that. We see that those flies are actually resistant to this long-lasting effect of sugar. So it tells us that there is actually some long-lasting effect of signaling through this pathway. Now that could actually be good news on a low sugar diet. What that might mean is that if you do this experiment the other way around and just reduce the activity of this pathway for a short period of time, that might actually have long-term benefits. And so that's exactly what we see here. So if we reduce the activity of the insulin signaling pathway for just a few weeks in early life, what happens? So looking here in red is our control where we reduce insulin signaling throughout life. And what you can see is this nice extension of lifespan. And these flies are actually living for about 1/3 of a year, which is a pretty long-lived fly. If we reduce insulin signaling for just these first few weeks of life, then actually we can see that just doing that actually still increases lifespan. It accounts for about 50% of the increase in lifespan that we see when we actually do this for the entire life. So bear in mind that these flies here are dying less, they're dying at a slower rate, even though we haven't done anything to them for several months at this point. So it's, I think, a really cool result that we have these long-lasting effects of signaling, which correspond to what we see when we manipulate diet. So that sums up really the final part of the talk here. So what we've seen is that diet and signaling do interact to determine aging. So we've seen that a dose of sugar reduces lifespan and increases body fat, and that sugar in just early life can actually shorten lifespan. But to really understand what diet is doing, we also need to understand how signaling and genetics are working. So we can actually increase the level of insulin signaling and RAS signaling, and that will actually protect a fly against these deleterious effects of high sugar. And we can do that experiment backwards as well. And we can see that reducing signaling just in early life is actually long enough to improve health in late life, which holds out some hope for maybe developing drugs that could target insulin signaling just early in life. So this brings us here to our final model. So we don't have all the pieces yet, but we've got some evidence that tells us that we really, in order to understand how our health works throughout our life, to understand obesity and aging, we really need to understand not just how our gut bacteria or our diet or our genetics are actually working. We really need to understand how all three of these work together. And if we can do that, then we might actually be able to predict what will be the best way of improving health. And we can do so in a way that is hopefully affordable and equal. So it just remains for me to thank my colleagues. There are far more people who worked on these things than I have space for, but I'm just putting here the people who actually produced data which has gone into this talk. I'm running my own lab here now at the University of Glasgow, but some of this work was actually done in labs where I formerly did postdocs, and so I'd like to acknowledge those former supervisors and advisors, and former funders and universities where this work was done. So with that, I will wrap up, and I'd love to take any questions. Thank you very much.
Excellent. Thank you, Adam. Appreciated. So questions. Let's start with a question from Mohammed Jamai. So from a technical standpoint, how do you remove the microbiota?
That is a really good question. So there are a couple of ways of doing that. The way that we do that here in my group is by decorionating the eggs. So basically the bacteria sit on the outside of the egg. We can take our fly eggs and we basically just drop them into 10% bleach solution for about 6 minutes. And then we wash them with sterile medium, sterile water. And we put those sterile eggs into sterile food, and then those flies grow up without bacteria.
Cool. And from Nicholas Insane, in which cells do you manipulate the signaling? So what cells do you think you're manipulating signaling? You can speculate where these signalings take place to impact lifespan. So where in the animal?
Sure, that's a really good question too. You can actually manipulate lots of different signaling pathways in different places, and that will have an effect on lifespan. And we think that that is probably because different things go wrong in different places as an animal ages. So for example, if the thing that maybe goes wrong in your gut that might cause you problems in age, probably going to be very different to the things that go wrong in, say, neurons as you age. So all of the experiments that I showed you there, we were mostly looking at the fat body and the gut. So the fat body is obviously the fly's kind of adipose and liver-like tissue. The gut is kind of my favorite tissue. So I'll be honest, that's kind of where I tend to go. But I have colleagues who have manipulated actually some similar pathways in neurons as well. And you can see similar benefits to lifespan of reducing insulin signaling in those places.
Cool. Wow, questions are coming in fast and furious, so this is good. So we'll get to as many as we can.
Okay.
Actually, there's one question that came from multiple people. I don't know if you can answer it, but others, such as you, Sophia, can maybe weigh in on this. From Abdullahi Hussein Umar, and everybody's going to forgive me for not pronouncing these correctly. I'm from New Jersey. And Solu Atunwa, which strain of Drosophila survive best in tropical regions? It seems to be something of real interest here in this group.
Yeah, I'm afraid I actually don't know the answer to that question. So there is some slightly older literature which has looked at the effects of certainly of climate, of temperature on lifespan. I do not know of a paper where they have actually identified a particular Drosophila strain which does better in tropics or tropical-like conditions. So,
does anybody else want to weigh in on that? Because this is a question that's come up multiple times. Okay, how about a question from Lola Martin Bermuda? Does diet impact the microbiota?
another excellent question. So this may be something actually which our other speakers have a slightly better grasp on than I do. Off the top of my head, I cannot think of a study that has done that in Drosophila. It definitely corresponds to the microbiota in other systems. So in mice, and humans as well, there is very strong correspondence between diet and composition of the microbes. And actually, there is some really cool stuff in humans where you find what are called enterotypes of bacteria, where the overall community of bacteria that people have is actually very strongly predicted by diet. So you can kind of take a random sample, look at it and say, yeah, that's probably someone who eats a lot of meat. That's probably a vegetarian. So yeah, it seems likely in Drosophila, but I haven't myself gone out and measured that.
All right, cool. And Sophia reminds me, but now I'm going to remind her that I asked her to weigh in about, she provided the answer to which Drosophila work best in the tropics on Slack. But, you know, I'm going to defensively hit back Shadow one. So did you want to say a couple things, Sophia?
I just answered the question, and regarding growing Drosophila in a lab, I think you shouldn't have any problems with Drosophila melanogaster. There's a reference that I just put in, which is a study that actually studies different Drosophila species at different temperatures, if you're interested in having a look at it. It's in the Slack channel that we've opened for today. Okay?
Cool. Thank you. All right, let's see, where do I start? Mariam Salaudin says this presentation is very interesting and comprehensive, which I would agree. It's always good to start your question with a compliment. It would be really cool if the insulin pathway could be manipulated early in life so we can enjoy all calories in whatever form they present. First of all, I wholeheartedly agree, considering what I just had for lunch. And secondly, and that is true, and secondly, what about this? What is known about humans and you know, dietary restrictions and so on? It's clearly in model organisms, you cut calories, they live longer. What about in people?
Yeah, so that's obvious. I mean, that's kind of the $1 million question, right? So I will plug a talk by Linda Partridge tomorrow. So she is a former mentor, and she's very interested in taking these things that, you know, she's been doing in Drosophila for an order of magnitude longer than I have in terms of years. She's really interested in taking some of these things we're finding in Drosophila and asking if we can actually apply them in humans as well. So I'm going to say that she's probably the right person to really give you the answer to that question. So there are, but I will go and I'll try for now. So there are kind of anecdotes, there are kind of randomized control trials which are underway to see whether some of these reductions that we see in feeding can actually influence human health. For the longest time, these were just anecdotes where maybe it was just 1 or 2 people who have just decided to, you know, reduce their own calorie intake. The biggest problem with these interventions in people is actually compliance. That it turns out people quite like eating nice food. And so, you know, people will say, yeah, sure, I'll sign up for your trial. And then a few months later, the clinician checks in with them and nah, I've stopped. I got bored of that, you know, I like my lunch. So I think the jury is still out. One of the more promising ways that we might get around that problem is through an intervention of intermittent fasting, which is where you don't reduce your overall diet completely throughout, But actually there do seem to be some benefits of maybe just taking a day or two a week where you don't eat very much. And so, yeah, the jury is still out, but I think we'll hopefully know in a couple of years.
All right, cool. Well, we have a blizzard of questions. So I'm going to only ask one more because we have to move on. So thanks everyone for those questions. Just drop those questions into Slack, and I'm sure Adam will be happy to weigh in on those and actually maybe have a little back and forth.
I'd be
A
delighted.
little community discussion would be nice because the chat section, as you can see, is pretty overwhelmed.
Sure.
Good. So let's see. Last question is the next question. Jojo Oyebo. We understand the protein is good for children. Does it really apply to flies? For example, increasing protein in young flies and then reducing sugar as they age.
Again, I don't think that switching experiment has, to my knowledge, been done. That's a great question. Someone should go do that. I think as we start to unravel this process, it is going to be, I think that question recognizes a really important point about translating these findings, which is that we really need to understand what is good for you at one point in life and bad for you at another point in life. And to be honest, it's actually that kind of trade-off that really interests me the most. It's trying to understand why it is that something which is good for you at one point is bad for you at another point. And if we're going to actually apply these things and translate them medically, I think that's going to be one of the really key things to address. So great question.
Yeah, I agree. All right. Actually, this is really good discussion, and already I see some projects emerging from this.
Absolutely. Yeah.
So, all right, but we do need to move on because I've actually been really looking forward to this. So first of all, thanks, Adam. Much appreciated. I really think the work from your group is amazing, and I can see where a lot of this is going. So good luck, and I'll check in behind me. Adam lives right behind me, as we just found out.
And thanks everyone for listening and for the great questions. It's been really, really stimulating. Thanks.
Definitely. So